Welcome to the next video in the Patterns in Nature topic. This video is going to be looking at the dot point, use available evidence to perform a first-hand investigation and gather first-hand data to identify and describe factors that affect the rate of transpiration. So again, it's a first-hand investigation, so we need to carry out some kind of experiment and we need to be able to gather data ourselves that leads us to be able to identify and describe the factors that affect the rate of transpiration. So the aim of this investigation, obviously, to be able to meet the syllabus dot point, is to identify and describe factors that affect the rate of transpiration, not respiration, sorry. So just a bit of an introduction, just to get us up to speed, all on the same page on where we're headed. Transpiration is the evaporation of water from the leaves of plants. Okay, so obviously evaporation is the changing of liquid to a gas. So this is exactly the same, but it has to be from the leaves of plants. Okay, so when plants undergo gas exchange, as we know, we've talked about the stomata opening. So when the stomata opens to allow gas exchange to take place, we've already mentioned that the issue that takes place is water also moves out of the leaf. Okay, so if a plant's already in water stress, opening the stomata for gas exchange is going to cause the plant to lose more water and obviously going to suffer. So it is the role of the guard cells to ensure that the plant has sufficient water to be able to carry out gas exchange without suffering from water loss. Because if the plant loses too much water, then the cell wall comes away, or the vacuole wall comes away from the cell wall, the cells begin to wither, and then the whole plant begins to wilt, and basically the plant begins to shut down and die. So some of the factors that can affect the rate of transpiration are temperature, wind, humidity, having hairy leaves or waxy cuticles, the surface area of the leaves, and a number of stomates. So the higher the temperature, the higher the rate of transpiration. Obviously, if transpiration is evaporation, just like any situation, if you increase the temperature, water evaporates faster. The next one is wind. So the windier it is, the higher the rate of transpiration. So obviously, as the wind comes by, it removes the water particles from the surface of the leaf, allowing more water to be lost from the leaf itself. Humidity, the more humid it is, it actually lowers the rate of transpiration. So humidity is the level of moisture that's in the air. So if there's already moisture in the air, the leaf realizes that it doesn't have to undergo the process of transpiration, so transpiration rate lowers quite considerably. Hairy leaves or waxy cuticles also lower the rate of transpiration. As we know, waxy cuticles help to decrease water loss by forming a film over the surface of the leaf, so obviously the water can't get out. And having hairy leaves, a bit like when you're cold, they form little air pockets, so the water gets trapped in the air pockets and therefore lowering the rate of transpiration. The surface area of the leaves can have an effect on the rate of transpiration. If the surface area of the leaf is bigger, so if it's broad and flat, there is obviously more surface area for transpiration to take place, so the rate of transpiration is higher. Leaves that occur in warm, dry, arid conditions are usually very long and thin, so they don't undergo transpiration too often in order to keep that rate down, in order to obviously keep that water inside the plant. And lastly, the number of stomates. So if there's more stomates, there's obviously more pores for the water to be leaving the plant, so therefore there's a higher rate of transpiration. So what we're going to do is we're going to set up this investigation that looks at a couple of different factors that we can reproduce in the laboratory and have a look at how they impact on the transpiration rate of leaves of geranium cuttings. So what we're going to do is set up 6 equal test tubes with equal amounts of water, a 1 centimeter layer of paraffin wax. So the paraffin wax basically stops any water evaporating directly out of the test tube. So this ensures that any water that is lost from the test tube is lost directly through the leaves of the plant. Okay, we're going to have our experimental control. This question came up in class about what is an experimental control. So the experimental control has had all the controlled variables added to it, but no independent variables. So this is our base point. This will show us that everything that's happened in these other 5 test tubes is a result of our independent variable, which are the different conditions that affect the rate of transpiration. Okay, we're going to draw a line on the test tube to show the level of the water so we can see how much that's changed. And we're also going to weigh all of these before we begin. Okay, so at the end, we're going to calculate a percentage weight loss. So one of these test tubes is going to be placed in front of a fan, which obviously replicates increasing wind. One's going to be placed under a bright light, which increases the amount of light the plant is exposed to. One's going to be placed in front of a convection heater that's going to obviously increase temperature. One's going to be placed into a bell jar and placed on the windowsill. So what should happen is that should increase the humidity inside the bell jar as the water is going to evaporate, so transpiration will take place, but the water will then collect on the surface of the bell jar and basically create a little humid environment inside the bell jar. The last one's going to be set up with a bell jar, but with some silica gel. And what that should do is it should remove the humidity from the air. So silica gel works to remove moisture, which is why you get it in boxes of shoes when you buy new shoes, so the shoes don't go moldy before you buy them. And that should remove all of these other, or in particular, get rid of the wind and just see how just sunlight without any other impact affects the rate of transpiration. So just to recap our variables, our independent variable is the conditions that the plants are subjected to. So each plant is under a different condition. The dependent variable is the rate of transpiration, which will be measured by the amount of water lost from each of the test tubes. And some controlled variables, we normally say we want at least 3. So the amount of water in each test tube, the amount of paraffin oil placed on the top of the water in each test tube, the number of leaves on each plant cutting. Obviously, if transpiration is the evaporation of water from the leaves, if there's more leaves, there's going to be more transpiration, and the time that the plants are left for. So we're going to leave them set up for a couple of days, come back and take our final readings. So when you do that, you're going to calculate the percentage weight loss. So we need to have our initial weight and our final weight, and you'll find the difference. Divide that by our original weight, and to turn that into a percentage, we'll multiply it by 100. Then you'll be able to gather results from another 2 groups and hopefully be able to calculate the average percentage weight loss for each of the 6 test tubes. Then to meet the dot point, all you need to do is be able to identify the different factors that affect transpiration, which we've done in this video. And from your results, you'll be able to see which of those different factors has the highest impact on the rate of transpiration based on the test tube that has lost the greatest percentage amount of weight. Okay, and then you'll be able to describe why you think that has taken place, and you will have met the requirements of this dot point. And that brings us to the end of this video, so thank you for watching.